Thank you for joining me here at PacBio Global Summit 2020. My name is Jenny Ekholm, and I'm part of the PacBio team. I will present on how HiFi sequencing can help increase solve rates for rare and Mendelian diseases. There are many types of variants in the genome, ranging from single nucleotide variants, deletions, insertions, tandem duplication, interspersed duplications, inversions, translocations, and copy number variants. These are all very important because that is what makes us different from one another and what contributes to both health and disease. Even though single nucleotide variants are by far the most common types of variants, due to their size, they still only make up about 5 megabase of the human genome. Aside from the CNVs, 3 megabases of the human genome is made up of indels, and due to the larger size of the structural variants, they actually make up 10 megabases of the genome. In order to detect the smaller variants like CNVs and indels, high accuracy is needed, and therefore these traditionally have been detected using short-read sequencing. For larger structural variants where the read length is key, long-read sequencing has been applied. However, with HiFi reads, you no longer need to choose between read accuracy and read length. You can now get highly accurate long reads that can discover all variant types ranging from CNVs to large structure variants. Not only can HiFi reads capture all variants, but they can also detect variants that are often missed with short reads. Here's an example of a structure variant. On the top, you see PacBio HiFi reads, and on the bottom, short reads for the same sample. HiFi reads are long enough to span the entire region, and therefore the 1700 base per deletion can clearly be detected, whereas the short reads are not mapping well to the region because of an underlying repeat expansion. In addition, HiFi reads enable phasing of the alleles, confirming that it's a homozygous deletion. Even though short reads are commonly used for CNVs and indel detection, they still miss a portion of them, especially the ones that are located in difficult to map regions of the genome. In a recent publication in Nature Reviewed Genetics by Larson and colleagues, they concluded that short reads miss 47% of the deletions and almost 78% of the insertions, and this was after applying 11 different variant callers specifically designed to detect these type of variants in short read data. Here's an example where CNV is missed by short reads. The STRC gene, which is known to cause congenital deafness, can easily be sequenced and alleles phased using HiFi reads, here shown in the middle. However, on the top, Illumina short reads have large gaps, and on the bottom, ONT reads have many errors. The reason for the difficulty in this region is that part of the gene, here indicated with arrows and dashed lines, has a pseudogene copy that is very similar in base context, causing mapping errors between the pseudogene and the functional copy. Overall, it's been shown that NGS short reads miss about 5% of the medical exome. This might not sound like a big number, but it affects 192 medically relevant genes. However, with the higher precision and recall, HiFi reads are able to resolve most of these regions. In order to generate highly accurate HiFi reads, every molecule will be sequenced at higher coverage. The sequencing polymerase has strand displacing capability and will therefore open the SMRTbell into a circle, allowing for the sequencing of both the forward and the reverse strands. The sequencing reaction continues until the sequencing run has completed. The sequencing errors, here indicated as red dots, are random and therefore will be washed out by increasing the number of passes around the SMRTbell. The consensus accuracy for a HiFi read with 10 passes is 99.9%. The HiFi sequencing workflow starts with 9 to 10 micrograms of input DNA that is shared down to 25 kb pieces. Adapters are then ligated to each end of the double-stranded DNA to form SMRTbells. Next, the sample is site selected, which means that only SMRTbells over 15 kb will be selected for sequencing. The library preparation can be completed in 1 day, and the HiFi library is then sequenced on the Sequel IIe system with a 30-hour movie time. Because this is a single molecule sequencing technology, the sequencing data will have a read length distribution. As seen here on the left, Most of the reads will be around 15 to 16 kb for a HiFi sequencing library. The median accuracy of the data is over 99.9%. This means that if you have a read that is 19,820 base pairs long, then you will only have about 8 errors in the read, indicated here as red dots. Therefore, the read is highly accurate at a Q value of 34. Because the reads are so accurate, they can be applied to standard bioinformatic pipelines that would be familiar to most of those who have done short read sequencing data analysis. The first step is to align the HiFi reads to a reference genome and then call the variants with tools like GATK or deep variants. These tools will produce a VCF file that contains the sequencing data, which can then be phased and visualized with tools like WhatsApp and IGB. Even though rare diseases are individually rare, they make up of almost 400 million cases globally, which means that 1 in every 10 individuals are affected with rare disease. For diagnosing these disorders, the initial test is whole exome and whole genome sequencing. However, over 50% of the cases still go unsolved. Therefore, the big question is there any way that we can increase the solve rate and help these patients to end their long diagnostic odyssey? This historical slide demonstrates how solve rate for rare diseases increases with technology evolution. The higher the genome resolution, the more comprehensive you can detect variants in the genome. With chromosomal karyotyping, 5% of the rare disease cases were solved. With microarray technology, that number increased to 12%. With short read sequencing, the number is about 30% for exome sequencing and below 50% for whole genome sequencing. For HiFi sequencing, that number is still undetermined. However, there has been numerous cases already where HiFi sequencing has been able to solve previously unsolved cases. So Matsumoto and his team published a paper in 2019 outlining the general study design framework for rare disease. First, if you have a strong candidate gene, then you start out with a targeted assay. However, if you have multiple candidate genes or no strong candidate genes, then you start out with whole genome or whole exome sequencing. And if these assays are uninformative, then reflex to PacBio whole genome sequencing in order to see the hidden structure variants and repeat expansions. However, this recommendation was before HiFi sequencing, and as you can see from this comparative table, HiFi has the same precision and recall for CNVs as short reads and much better capabilities to capture a larger structure variants. Therefore, a more affirmative strategy is to start directly with HiFi sequencing, either to targeted sequencing if you have a strong candidate gene, or alternatively, if not, then start with HiFi whole genome sequencing to capture structure variants and repeat expansions in addition to the small variants. Numerous groups have applied this approach to rare disease for cases that have gone previously unsolved. And as you can see here, there's quite a few publications already out. And I will now go into a few of these studies in further detail. First, Kristen Soon from Cincinnati Children's Hospital applied smart sequencing to 2 rare disease cases with neurological symptoms. These had gone unsolved despite a large panel of genetic testing, including microarrays and short read sequencing. The first case was a 9-year-old with short stature and hypotonia. In addition, the child had symptoms of developmental delay, nocturnal epilepsy, and speech delay. They applied low-fold whole genome sequencing to the sample, and they detected over 31,000 variants. After filtering, they were able to narrow that down to 6 variants. These 6 variants were involved in complex rearrangements on chromosome 2 and chromosome 4. One of these structure variants of chromosome 2 landed in the MBD5 gene that included changes in the protein that regulates the gene's transcription. This gene has been linked to developmental delay, speech impairments, seizures, sleep disturbances, and autistic-like behaviors in a condition that is known as the MBD5-associated neurodevelopmental disorder, or MAND. These results were confirmed with Sanger sequencing, gene expression, and optical mapping. The second case that Kristen soon looked into was equally complex. The patient was a 70-year-old with chorea, myoclonus, anxiety, hypothyroidism, and a family history of early death due to lung cancer. Again, she applied low-fold whole genome sequencing to the sample and found over 29,000 different variants. After filtering, she was able to highlight 8 different variants, all on chromosome 14. Altogether, she discovered 5 breakpoints, some upstream and some downstream of the NKX2-1 gene that previously been reported to have pathogenic variants. The work is still ongoing to pinpoint the structure and mechanism of this finding. In another study, Dr. Matsumoto from Yokohama City University studied a sibling pair that both showed symptoms of epilepsy. However, the disease-causing variant had not been identified with whole exome sequencing. So they applied smart sequencing to 1 proband and 3 unaffected members that served as controls. After filtering, they narrowed down the candidate variants to 2 deletions and 3 insertions. They found a 12.4 kilobases homozygous deletion spanning the first coding exon of the CNL6 gene. They hypothesized that this deletion had not been captured by previous technologies due to the high GC content in the region, which was 77.6%. In addition to these individual studies, there has also been some large consortia that are planning to implement PacBio long-read sequencing to increase the solve rate for their rare disease cases. For example, Solve R&D, funded by the European Union, is committed to applying PacBio long reads to 500 individuals. In the US, there is 2 large consortia applying HiFi sequencing to their studies. First, we have Children's Mercy, Kansas City, who are applying HiFi sequencing to the Genomic Answers for Kids project that is planning to sequence 100,000 individuals total. And Discovery Life Sciences at Hudson Institute of Biotechnology has started to apply long-read sequencing to thousands of human genomes to characterize both common and disease-causing variants. So far for the CSER project at Hudson Alpha, they have applied HiFi sequencing to 6 of their unsolved cases and were able to solve 2 of them. In a preprint, they referred to HiFi sequencing being a powerful frontline tool for research and clinical testing within the rare disease genetics community. To learn more about this study, please listen to Jeremy Smutz's Global Summit presentation titled HiFi, Amazing Applications in Plant Genomes and Human Disease Research. It can currently be found on demand. Another consortia project in the US is the Genomic Answers for Kids. Their aim is to generate a pediatric data repository to facilitate the search for answers and novel treatments for pediatric genetic conditions. Their plan is to enroll 30,000 children and their parents over the next 7 years. Their testing pipeline starts with exome sequencing and whole genome sequencing using short reads and also additional functional genomics. For the unsolved cases, long-read sequencing is applied. Emily Farrow from Children's Mercy in Kansas City gave a status update on their progress so far during the ASHG workshop talk. They selected 3 unsolved cases and applied 30-fold HiFi coverage to these samples. The first case is a 4-year-old girl that was diagnosed prenatally with a disorder that still persisted after birth. The initial clinical NGS testing found a single pathogenic variant in the MPC gene, indicating Niemann-Pick disease type C. When this patient was included in Genomic Answers for Kids consortia, their analysis revealed a second intronic pathogenic variant. However, the phase of this variant was still unknown. Therefore, this sample was submitted for HiFi sequencing, which revealed that the pathogenic variants were in trans, which means that they're on the opposite allele, so they inherited 1 from each parent. The second case Emily Farrow and her team looked at was a child that had developmental delay and seizures. Subsequent MRI testing revealed lissencephaly. The sample was submitted for exome sequencing, however, the test came back negative, so HiFi sequencing was applied. which identified a pathogenic variant in the CEP85L gene. In the third case, they had a family of 9 affected members that showed symptoms of dystonia, seizures, and ataxia with variable degrees of severity. All previous tests had come back negative. 4 out of the 9 individuals were then sequenced using HiFi sequencing, which identified a pathogenic repeat expansion in the STRD7 gene that had also previously been linked to familial adult myoclonic epilepsy. So they also tested the remainder of the affected family members, as well as unaffected members and other controls. And what they found was that all affected members had the repeat expansion present, whereas all the unaffected did not. In summary, they were able to solve all 3 cases that previously had gone unsolved. They also found that over 450,000 CNVs and indels and 10,000 structure variants had gone previously undetected with other technologies. Therefore, now with the more comprehensive variant detection capabilities, they were now able to increase their solve rates. All in all, the general trend across all these studies is that HiFi sequencing has the ability to see more variants, and therefore it also has the ability to increase the solve rate for rare and undiagnosed disease.